So welcome everybody to another one of our seminar series. This is an initiative from Open Neuroscience together with Worldwide series, and this is all about open source tools, open science tools for neuroscience. Today we have the immense pleasure of having with us Professor David Kennett, who's going to be presenting about ReproNim. But before we start, I would like to say a few words about Professor Kennedy. He's a professor of psychiatry at the University of Massachusetts, the medical school, and he's the director of the Division of Neuroinformatics at the Child and Adolescent Neurodevelopment Initiative. He has extensive expertise in the development of image analysis techniques and was the co-founder of the Center for Morphometric Analysis, the CMA, at the Massachusetts General Hospital. His career has been in his career, he has seen participation in the advent of such technologies as MRI-based morphometric analysis, functional MRI, and diffusion tensor pathway analysis. He has a long-standing experience with development of neuroinformatics resources, and he is the co-PI liaison for the Neuroimaging Informatics Tools and Resources Clearinghouse, NITRIC. He is the PI of ReproNim, a Center for Reproducible Neuroimaging Computation. In addition, he's also the founding editor of the journal Neuroinformatics that debuted in 2003. So as you can see, he has an extensive career behind him, always working with open tools and neuroinformatics, and we're really, really happy to have you with us today. Thank you very much again. For people in Zoom, Professor Kennedy has agreed to take questions during his talk, so please do write them on the chat so that we can relay them. If you're on YouTube, we can also ask questions there, and we also relay them. So without further ado, Professor Kennedy, please, the floor is yours. Thank you very much.
Thank you so much for that kind introduction and the opportunity to participate in this seminar series. Quite looking forward to it. Most of the nice things you said about me in my introduction are mainly because I'm old and I've been around for an awful long time. But one of the good things about being old is, I remember when science wasn't open science, it was closed science, and a lot of things, data was not shared very well, software and tools were not shared all that well. So really the community over the last 33 years that I've been involved in it has really been making great strides, towards moving science, meaning open science, not science, assuming to be closed science. So as was indicated in my introduction, I sort of have three jobs that I do. The first one is I work in the Child Neurodevelopment Initiative, where we do studies on developmental disabilities, autism, ADHD, using MRI-based techniques to look at structure, function, diffusion, and try to understand the correlates of brain behavior, brain structure, and brain behavior. The second job is I co-run NITRIC, the Neuroimaging Informatics Tools and Resources Clearinghouse, a one-stop shop for people who need to find software, open or closed, or find data, and find procedures and find a community, to work with. It's also one of the hallmark places in neuroimaging that if you're developing software or collecting data, it's one of the opportunities, one of the platforms to share that information with the community. And then the third project I work with is ReproNim, this NIH, NIBIB P41 center, to really try to get to the last mile of reproducible techniques, to really try to make an impact on the neuroimaging community and the way we observe and publish findings. I semi-apologize for the talk being kind of neuroimaging centric, but every issue and topic I bring up is not really unique to neuroimaging. It's just the place where we try to deal with it most. I hope that if you're from a different specific field, you can hear what I'm saying and see where that maps on to what you're doing, and hopefully, and that's a great dialogue place as well, to discuss if needed. I used to start this talk with a very polite and gee, wouldn't it be nice if we could all do these good things? But I've recently gotten old and mean and decided, no, let's not beat around the bush. Reproducibility, just do it or else. you won't get funding. There are new initiatives coming down, the pipe from many funding organizations that really are turning up the importance of proper data management and proper data sharing. And a particularly new large effort is going into effect with the NIH, January 25th, 2023. So if you don't get ready ahead of time, you're going to be behind and behind the ball in terms of doing the types of things the funding agencies are going to make you do. At least within the NIH, and maybe true with other funding organizations, is the annual progress report is getting pickier and pickier about the data sharing, the data collection, the procedure sharing, and it's coming closer. And with the new policy, you may not get year two's worth of money until you've done your obligations for year one. And also with these new obligations, your next grant may be judged to some extent on how well you accomplished your data management in prior awards. So you won't get funding, you won't get published unless you start worrying about this. Many journals have software and data sharing policies. So again, that's easier to take care of if you're managing things well from the start at home, as opposed to being a hurdle that you have to do between the acceptance and the publication. If you can't get funding and you can't get published, you won't get tenure or a job promotion or that next school job. And things like that, even if tenure is kind of passé these days, but it'll be bad for your career if you're not published and you're not funded and you're not taking these kinds of mandates seriously. And most importantly, John Oliver will laugh at you. Now, again, I'm not going to play the John Oliver YouTube, but there's a link to this that I'll make available. But he has a field day making fun of science and reporting of science and how irreproducible science is, and that's just embarrassing for all of us. So the key is to be reproducible by design, not reproducibility as an after effect. You really need to embrace as much of these concepts as possible. And again, there's some tools that help make those concepts more accessible, but think about it from the beginning and think about it at the end. Okay. So in my little time with us today, I will say a little bit about trying to motivate and give an example of the existence of the reproducibility problem. To some extent, most people who hear these kinds of talks are fairly aware that that such a problem exists. But just in case, I'll give one example, and that's just many. We could spend a whole seminar talking about examples of the reproducibility problem. The causes of reproducibility problem is that everything that you do in a huge complex experiment can make a substantial difference to the exact outcome you get. And again, when we're looking at subtle things, details about everything can matter. Then you'll be introduced to some general guidelines and framework about how we try to address the reproducibility problem, sort of theoretically, and we'll look at some the specific tools and resources that may be helpful to make you and your work more efficient and less irreproducible in your ongoing efforts. Reproducibility has been a problem for a long time, and it manifests itself in many different ways. There are publications that lament how badly drug companies do at taking basic science leads and turning those into actionable medical compounds. And often when you dig back, the scientific leads that are driving drug company discovery are often flawed and not reproducible themselves. So if we the basic science are feeding the drug companies bad leads, then of course that's inefficient for them to develop an outcome product. So there's examples of that. The one I happen to lean back on, particularly in mental health, the mental health topic, was a comment by Tom Insel, the head of the National Institutes of Health Mental Health Division, where he was lamenting back in 2012 that there's really a profusion of statistically significant findings. We get lots of publications. There's no lack of things that we publish, but these significant observations tend to be minimally differentiating in terms of the biologic interpretation of them. They only support approximate replications in sort of superficial ways that makes it very hard to either confirm nor deny a particular observation. So there's been 30, 40 years of very expensive, neuroimaging studies. There's been 30 or 40 years of very expensive genetics studies. But today, if your kid with autism or your adolescent with a drug issue or your adult with schizophrenia walks into their psychiatrist's office, the prognosis and the interventions are pretty darn similar to they were 30 years ago before billions of dollars have been invested in various research methods. Now, again, that's slightly overstated. There are some, success stories, but not nearly, as much as we need to be. The causes, for this, kind of dearth of findings, dearth of, sort of repeatable and actionable findings, have to do with, many studies that are, low power, small n. There's an issue with sort of what is it we're targeting. The diagnosis, autism may actually be a bad target in the sense that autism is a very heterogeneous type of thing, and it may not be a specific finding that links all of autism or depression or even schizophrenia together. And so that we really need to be looking at a more specific domain of function or domain of behavior to be the target of what we're looking at. And important issues in that we write papers, but the papers we write really are incomplete in terms of the methods and incomplete in terms of the results, and leave too many places for misinterpretation or differences to creep in when one tries to understand, replicate, and generalize the literature. And finally, because we have this predilection to publish positive, significant things, we have this publication bias, and that things that aren't confirmed rarely show up in the literature. So we really miss all of the sort of negative, destructive interference that we'd expect in a normal environment to really be able to beat down some spurious false positive findings by the lack of the negative findings that go with them. So all of these feed in to a literature that is full of billions of results, and those results aren't particularly well described. They're often flawed and have false positives or other statistical issues, and then we're only seeing sort of the positive tail of those distributions when we look at the literature. And so all of these are things we can do something about, and we just need to be aware of this and cognizant of it. So an important question, to start with is, to reproduce what? What are we really talking about when we talk about reproducibility? And you'll see lots of different people talking about reproducibility. We'll talk about different, definitions, of things. And so I'm going to throw a couple concepts out here. We first have reproduce in the terms of re-execution of a particular result. I would certainly fail this challenge, but I challenge many of you to go back to a paper from a couple years ago and find a particular result and find that data and be able to generate that same result de novo. We just tend to not necessarily operate in a way that challenges that and exercises that effort. But again, we really need to gain control over the re-execution of our primary findings. So if I have a particular neuroimaging data set, I have a particular tool, FreeSurfer, that generates volumes, of the hippocampus. Can I go back to my collection of data and next year, confirm that the volume of the hippocampus is 203 millimeters larger in the boys than the girls at some particular observation. If that's what I published 5 years ago, that is really something I ought to be able to regenerate that finding. And if I aren't able to do it, then others wouldn't be able to do that as well. So this concept of re-executability of the primary findings being able to be re-executed is one of the core pillars of the reproducibility definitions that I want to lay out here. But then the other, that's not really the end goal. I don't really care about the volume in that particular study. I want to generalize that finding. Is it true that hippocampi are larger in boys than girls, regardless of the tool, regardless of the exact data? So that's where we get into this reproducibility, the reproducing being the issue of generalization of a claim. Can we take my first re-executable observation and generalize it by doing it on more data, doing it with different tools, and still identify that hippocampi are larger in boys than in girls. So these are sort of the balances that, unless we have sort of re-executability at a very fine grain, it is very hard to really confirm the generalization of a claim, because it's very easy to, when you don't generalize a claim, just wish it away in terms of, oh, it's slightly different in the data, oh, it's slightly different in the tools, and so we find some different thing, publish that, and run along our merry way, and never go back to understand why a particular finding did or didn't generalize it. Was it power? Was it the specific tools? Was it details of the population and things like that? And things that don't replicate are very important, because we need to understand why. What is it about the tool? What is it about the data? What is it about the statistical, the modeling that I'm using? So since we have the re-executability and we have the sort of generalizability, it made me think of Monty Python Life of Brian, where we follow the re-execution or we follow the generalization. And so we have different techniques for those topics. Okay. So the cornerstone of reproducible research is transparency. And again, the assertion here is we're really not being particularly transparent in the methods of our publications to date. So we need to be more transparent. Transparency is achieved through annotating and sharing with others through publication precisely what operations were performed and on specifically what data, in a fashion that could be potentially be re-executed by someone in an attempt to replicate and then ultimately document the generalizability of the original finding. So that's really our aspirational goal, is to get people aware and armed with some of the right tools to really get towards a re-executable publication, so that the publication, and we'll talk a little bit more about this at the end, but we'd like a publication to be associated with its data, to be associated with its software and computational environments, to be associated with the statistics, to be associated with the results that it generates, so that the ability to take a given publication and really build on it in an efficient and appropriate way is really taken as sort of a primary scientific objective of publication. An example of reproducibility issues that I like to lean back on is a paper by Anne Lefebvre from 2015 or so, almost 10 years ago, that looks at a very common finding in the autism literature. If you go back through a meta-analysis of neuroimaging studies that look at the volume of the corpus callosum, a particular fiber bundle that connects the left and right hemispheres, you find ample publications and ample evidence that the corpus callosum volume is smaller in subject with autism than typical controls. And this little meta-analysis is shown here. And while 11 of the 14 observations document a significant result, looking at the meta-analysis as a whole is very clear that there is a diagnosis effect of smaller corpus callosum in children with autism. Fine, so that feels like that's exactly what the literature is supposed to do. If it's a true finding, it ought to replicate and it ought to appear in the literature with different data sets, with different methods, and emerge there. The problem is, if you actually then, so that was all sort of historical data. Then in about 2015 or so, the ABIDE, the Autism Brain Image Data Exchange, became available. A large set of 1,000 and some subjects, MRI scans with autism, without autism, became available. And so in addition to a meta-analysis one could prospectively do a mega analysis to see if this observation is really as clear as what that meta-analysis made it look like. We should be able to see that same thing in a prospective set of data. So this is 694 subjects, 328 of them have autism, principally male, and there's 366 control subjects in this. We use a specific singular tool, FreeSurfer, and we have this abide data set. So you can do an analysis, build a nested statistical approach to this, and you can find some fairly clear things. There's a very significant site effect. Okay, well, we've known that different sites have different sort of systematic differences, and so the recapitulation of a site effect was exactly expected. We can see our expected age effect. The older you get, the larger the corpus callosum gets, so there's a highly significant age effect in this data. We see a highly significant sex effect. We know that there's boy-girl differences in the sizes of brain structures, and this data set recapitulates that sex effect as well. But lo and behold, when we go to look at the group effect, is there a difference between autism and non-autism? We do not get a significant effect in this particular data set. Now, so that of course caused lots of consternation that gee, why is such a clear meta-analytic effect not replicating in a mega analysis representation. Well, there's a handful of reasons that the authors posit. The first of which is this study has a very high power because it has 694 subjects. The power is about 0.95 to detect a small effect. All of the prior studies in the meta-analysis were actually fairly small studies, all of which had very low power to detect an effect of the size that they were saying. Oops. So I have to remember all this. So the highest power in the biggest of those studies was only about 30% to detect an effect size of the size that they were reporting. So again, this gets us into this the low positive predictive values of an awful lot of the studies. And if the effect were really only as large as it was reported, there should have been much more negative findings, and that given the low power, more of those publications must have existed that didn't get published that showed that small effect, just because if the effect were that small, it wouldn't show up that often in that particular type of observation. The other issue here is sort of covariant. There's a lot of things biologically that go together. The total brain volume is affected by the disorder. The corpus callosum is related to total brain volume. IQ is related to total brain volume and structural sizes. And so the purely looking at diagnosis effect and not properly accounting for IQ and total brain volume and other covariates can make it hard to detect once you solve appropriately for these other things exactly what the effect is. So it's not really that there isn't a diagnosis effect. There is an IQ effect, and that IQ effect is driven in large part by diagnosis. So depending on how you do the modeling, you can affect sort of how that comes out. And once you get into these larger data sets and the ability to more appropriately design models, it again changes the ability of things to be detected. So as this kind of alludes to when it comes to examining the re-execution or generalization of an observation, it turns out that everything matters. Some of this sort of seems silly. What operating system your computer is matters. If I take some FSL, the FimRib software library, to generate brain structural volumes, and I run that same program on my Mac, and I run it similarly on a Dockerized Ubuntu or a bare metal Ubuntu, I can get differences in my results numerically based on the operating system. And there's a lot of examples of these kinds of things. The same tool, the same version of a tool, does not behave the same in different operating system environments. data matters. If you have lots of subjects versus a few subjects, you can imagine, answers, can change. Here again, we're looking at the hippocampus volume, the sex effect. Back in 2005, the NIH pediatric data set became available, 325 subjects. Looking at that sex effect, we end up sort of not seeing a significant effect. Fast forward to 2021, the PING, the Pediatric Imaging Neurocognition and Genetics study becomes available with 1,200 more data sets. And lo and behold, now that we're up to 1,500 data sets, this observation begins to resolve itself. And then later we add the ABIDE and other data sets, and this particular feature can remain. So again, as time changes, as we get more power, as we get more shared data, that can make a difference. That a publication in 2005 may have said no significant difference. These subsequent observations are within the range of that original observation, but we've now narrowed down the error bars on this and begun to have a sort of a stable observation there. So again, depending on the effect size, depending on how large an effect we're looking at, it really can be surprisingly large numbers of subjects that are necessary in order to resolve some of the finer grained observations that we're looking for. This just illustrates that in a very specific way. This if this is doing permutation tests, this is using the PING data set. It's 1,200 subjects. If we take a permutation of 1,000 of those subjects and look at the hippocampus sex effect, regardless of what subset of 1,000 I take, I see a fairly stable observation of about 300 or 250 cubic millimeter effect size. As we use smaller subsamples, eventually we get down here to subsamples of that where we begin to have numerous subsamples of these where you're not seeing a significant effect. So now this is purely recapitulating the effect size, the N, and the power to observe a particular effect. So it turns out that this hippocampus gender effect really requires samples on the order of 250, 300 in order to generate that stable effect. And in general, in the older school imaging data, we are very rarely working with sample sizes of that size. And even this is just defined a medium effect size result. So again, I think we've been deluding ourselves a little bit about the types of things that we see in our smaller underpowered studies. One of my mantras, I don't think it made it into this talk anywhere, is everything is underpowered. I don't care how many subjects you have. The biology that we're looking at is so complex. There's so many other things that you should covary for, that even if you have a large sample, you then can covary for the things, then you still end up being underpowered. Anyway, what tool you choose to use matters. The neuroimaging community is blessed with having lots of different tools that are available for different things. It's also cursed by having lots of different tools that are available for different things. I'm not going to talk about which tool is right or wrong, but I'm going to tell you that if the popular FreeSurfer tool gets used or the popular ANTS tools get used, different versions of FreeSurfer are fairly self-consistent, but the FreeSurfer to ANTS correlation of certain similar same measurements. In this case, the volume of the left caudal anterior cingulate, a cortical region that both of these systems measure. The correlation, while significant, is really surprisingly problematic. So the next question is fine. They don't completely correlate as well as you'd like. What's the implication of that? And that puts me into this position where I talk about Siegel's law, which states a person with a watch knows what time it is. So my watch in this case analogy is FreeSurfer, and the time analogy here is the cortical regional structures that are significantly different between autism and control subjects in the ABIDE data set. And my one watch tells me that these structures here are this tip of the iceberg of the regional differences between those two patient populations. Great, one watch, I know what time it is. But a person with two watches is never sure. So my second watch in this case is ants. So the set of regions, that was, the hallmark of the types of regions that were, being given to me by, one tool. Free surfer is really quite different than the set of observations that are most significantly different between the populations with a different tool, ants in this case. And so that's the ramification of that sort of weak correlation between these measures that you can end up with fairly dramatic differences between groups. And while I highlight this one example because it's one we've done, there's been similar studies using schizophrenia and these tools, and again making it concerning that our answers are not really volume of the region specific. They are the tool that measures the volume of the region specific. And so again, that's important to keep in mind when you consider generalization and pooling observations across the literature. I don't need to belabor this, but exactly what statistical model you use matters. Again, I'm looking at my favorite little sex effect in the typical developing subjects. I can model this sex effect as a function of age and sight and socioeconomic status and brain volume and genetic ancestry. And I can look at this sex effect. It turns out if you don't, so the models that don't include brain volume give me a very large effect. But we already, since we're looking at sex and we know there's a large sex difference in male and female volumes, we really need to include brain volume as a covariate, and then we see our effect size reduces dramatically, but also our R squared, our ability to fit the data increases dramatically. So these are substantially better models that have better R squared in them, but they're also substantially reduced since we've taken out the large total brain volume effects and are left with a much smaller hippocampus specific brain volume differences. And then each of these little circles is a different permutation of all of these different covariates, all of which have historical reason to be included. We know age is important. We know there's site effects. We know that socioeconomic status affects brain volumes. We know that genetic ancestry factor affects brain volume. So there's no really necessarily good rationale to not include some of those, but some of these models that includes these things end up being not significant, whereas others of these models with very similar effect size end up being significant just based on the ability of these to model the underlying sources of variation. So again, I'm not necessarily here to talk about what the right model is. I'm just here to make sure that we keep in mind that exactly which model is critically important to what things do and don't appear significant and which things do and don't appear to generalize. So in our 5-step program to recovery, we have to admit first we have to admit that doing truly reproducible neuroimaging is hard. There are so many things you have to do in order to get it right. So ReproNim has been working to try to establish a comprehensive set of tools and practices that are designed to advance reproducibility in each of the critical stages of the neuroimaging research process. The conceptual goal that we're after, so here's an example of a traditional publication that's looking at the corpus callosum area versus brain volume in typically developing subjects, in this case from the 2005 pediatric NIH pediatric normative data set. Here we see a lovely significant correlation that shows that corpus callosum area and brain volume are coupled, although fairly weakly. That's great. This article, this figure appears in a PDF somewhere, and great. A ReproNim variant of the same concept shows a similar thing, and we've gone back to that pediatric data set. We've done it, measured corpus callosum volume and brain volume using modern tools, and we see this same effect. We see a lovely correlation between the corpus callosum volume and the total brain volume. But in the ReproNim variant of this, in the modern variant of this, each of these points really needs to carry along with it all of its provenance, all of the where did I come from answer. You should be able to go to any one of these points, that point via the modeling and the descriptions that we hold on to now, that point knows who the subject is, and therefore what the demographics and clinical variables were. It knows what MRI scan generated that volume. It knows what the scanner is. It knows what the acquisition details are, and it knows where that scan lives at some public repository. In this case, the Abide data set. That point knows what workflow generated it, exactly what software, exactly what hardware, and this point knows where it as a concept, a measure of the corpus callosum volume, fits in the ontology and the knowledge of where different parts and volumes and structures live in the constellation of types of measures that one can be consider. So all of this content is attached to that point, so that from that point you can get back to it. The particular workflow that generated this happens to live in a particular container that you can get at from GitHub or get at from Docker Hub, etc. The point can be regenerated by anyone, as long as you can get to the container and that data set. So clearly I, the author, can regenerate that point, but you, the reader, should be able to regenerate that point if you have authorization to get to that data, and you can access the computational container or workflow that generated that. So the importance of that is, one, it can be re-executed by anyone, but also when I have new data, I can make sure that I subject my new data to an identical process, and not some slightly different version of the operating system, or some slightly different version of the software, and things like that. We may want to explore what those dependencies are, but at least we can start from getting back to anyone can generate that same point in terms of the re-executability, which then supports my ability to say, if I have new data and I use the same processing, do I get data that falls on the same trend or not? Or is there something special about what I had done before? So this point we would say is fair in that it's findable, accessible, interoperable, and reusable. So if that is part of the publication, that point and its family of points and its family of statistics that belong to it, should be able to be found by anyone. That accessible, should be reusable by others, and interoperable. So not only are the points, fair and shareable and have their complete provenance, but the statistical interpretations, that particular regression, that we did, should be able to be regenerated, should be findable, should be extensible to new data, should be version controlled, and should generate the provenance that lets you track from that particular statistical interpretation to all of the details that went into generating it, so that can be regenerated and support the observations of generalization. So reproducible neuroimaging research is hard. There's so many things to do to get it right. So one of the main things ReproNim has had to try to do is to stratify the reproducibility processes into the individual components of research that are each less hard themselves and can be each adopted individually or gradually or incrementally, because it's very hard to get people to just jump whole hog into doing everything right from the middle of your study, or start new studies. We don't really want to wait for that. So there's lots of things anyone can do. Some are easy, some are hard. We want to give you a virtual path through these components, try to illustrate where easier things are, where harder things are, in order to improve. Today's paper is a little bit better than last year's paper, and next year's paper will hopefully be a little bit better than the paper than this year's. So just reminding ourselves of these general processes, a workflow entails acquiring subjects and acquiring imaging. We generate lots of imaging data. We subject this to some particular analysis workflow. We generate some derived data, statistical maps or volumes of regional or diffusion types of things. We apply statistical interpretations of those and make some conclusion and some claim that comes from that observation, and then we publish that. So these are kind of our stages of workflow. Each of these are an opportunity to make sure we annotate everything, and each of these are really publication options, publication broadly, in that all of these elements should be accessible to the final publication, And each of these themselves may be separate publishable entities along the way. So we'll go through the details of this a little bit in a bit, but again, so our phases really are associated with the study design phase, the data collection phase, data processing phase, statistical analysis phase, and the publication phase. And each of these phases of the research effort, we want to be able to think about what is it you can do that makes this bit a little bit more reproducible, and which of these are low-hanging fruit, and which of these are harder to adopt, and therefore require more planning and processing. So across all those phases, we like people to consider these a number of approaches. First of which is we want really to plan everything in advance. One of the biggest problems I think the community runs into, and I run into it. So anything I complain about the community really means problems I've had in my career doing things. So often problems have to do with not planning things in advance. You get to your statistics, and the original statistic you're going to do doesn't really get to the thing that you want to do, so you change something. So find that adds to variance in the approach. Version control everything. We often, only passively, sort of write our acquisition protocol, and oh, we need to change something, and we just, change it, and we don't know how that, acquisition or analysis plans have evolved over time. And version control applies to data. Version control applies to scripts and software and our actual, containers and protocols. And again, we kind of think of version control particularly for software, but really version control is our protocols. They evolve, they change at a particular time, and they change for particular reasons, and we tend to lose touch with that evolution of things. The next objective is to annotate everything. And so by annotation, we really need to make our observations so that others can understand and reuse these more easily. One of the biggest problems we have in data sharing is, people hate at the data sharing time to have to annotate all these things so that they don't have to answer questions to the end users. People don't share because they hate to answer all the questions that someone's going to have to ask them in order to reuse that data. Well, if you annotated it, then people won't harass you so much. Well, if you wait till the end to annotate it, you may not really remember all that well what the version was, what the measurement was, etc. So annotate up front so that annotation of everything, of your raw data, of your derived results, of your analysis platforms happen while you're doing it, so that at the end, when you come to share, there's no thinking that even has to go into how to annotate, because you annotated along the way and you make that available. And really the goal for these annotations is to make the data self-describing so that no one should ever have to bother you about what it was that you shared with them. Using standards is an important mantra that again lowers the barriers to others using things. If you make up your own imaging format and then share that, it's going to be hard for people to know to use that, how to use that, and without you having to explain it to them. If you use standard formats, if you use BIDs, or if you use DICOM, if you use your standards, that really lowers the barrier to how people can access things without harassing you, the sharer. And the semi-interesting distinction here is standards for for computational environments are also one of our important topics here. And so that's kind of where these containerized computational environments fit in. So we can get a standard computational environment so that those can be shared, that others can access that same computational environment. So containers are a type of standards that apply to the computational environment. And publish, and again, publish broadly, somehow share, somehow make available to others, preferably with credit and credit to you, the author, but make everything available, the raw data, the derived results, the processing workflows, either as separate publications or at least attached to your primary publication, so that all the elements are easily attachable to each other. And again, the more you can publish and get credit for publishing separately the data, separately the analysis, you can actually get 3 lovely academic citations out of a study because you publish the data and get credit for that. You publish the workflow and get credit for that. And you publish the application and your conclusion and results from that, your claims from that. So we're throwing away way too many sort of places where the data and information can be shared, and credit, to the originators of these different things can be had. This is a horrible slide, but I wanted to take my someone taught me never to say a slide is horrible, but no, I did. This is our design, collect, process, statistics, publish, phases of the research process. This is that plan, version control, annotate standards, and publish procedural mantra that we want to get. So again, I was trying to flesh out here that there are a lot of tools, some that ReproNim is working on, and some that have existed and that try to help navigate this and give you tools to do a better job. This version control is kind of easy-ish. Git, GitHub, other version control systems are prevalent and are really need to be considered to be used. DataLad was originally called DataGit, is really Git version control for data. So it takes all the concepts of Git, happens to use Git Annex so that data can be managed in terms of versioning much more efficiently, and versioned data and computational environments through the ReproNim computational manager, ReproMan, can be help manage the version control of the processing. Containers are important for doing standard processing. NeuroDocker is a tool that we've developed to help make it easy for the neuroimaging community to create containers, since that tends to be a barrier. There were not standards for annotation for many of these derived products. So you see along here the NIDM, the neuroimaging data model experiment, neuroimaging data model results are sort of standards, provenant representations for ways to encode our behavioral, our subject, our result data in a standard that had to be developed, since there wasn't really a community standard for that. It builds on community standards as well. So there's standards built into that. Annotating data, the tools to annotate mean you need to apply these standard representations. So you need to apply these representations to your data as you acquire it, or to your results as you generate them. So there's a set of tools for managing that. All of these steps are generating provenance, and we need to maintain that provenance locally in ways that we're just not quite used to. And so that's where we get into this concepts of the repro ponds and repro lakes. As you go through your analysis, you're generating these NIDM representations. Those should be kept and kept in a common place. And from those data representations, one can track the provenance, the connection between observations and how it was generated, and the statistics, and then the pooled results from that. Okay, so the slides, and I'm going to make these available. I don't want to go into too much detail, but there's tools and ideas that can help make study design better, making sure that you get the appropriate consent. And I cannot tell you how many times even big national data sharing programs come to the end and, oh, I didn't get my subject to consent to sharing that data. Well, what the heck did you do in this big national consortium to collect all this data for if you didn't actually get the consent right in the first place? Anyway, planning data in advance, doing pre-registration, using standard markup and collection schemes are important elements in the study design phase, and make sure that you have the appropriate schema for the things you're going to do. If not, work with someone to help develop them so that no one else ever has to do that again. Data collection, bids, for the neuroimaging community, the neuroimaging data model together, really make it possible to completely represent our data sources using version control on our data, Git and our data processing between Git and DataLad, and annotate as you collect, making sure you apply annotation along the way, so that again you don't run into this bottleneck at the end. Data processing, be as modular as you can so that you can deduce later on when things evolve and change. Document everything through version control, and using standard workflow engines, using standard computational environments, use containerization, and there are places to keep containers for the community so that they can be reused forever, so to speak, using version control and, again, apply annotation as you're running your tools and not as an afterthought. My main point on the statistical analysis thing is that almost all of us don't know as much statistics as we should, and so we should all just keep working with our local statisticians and keep the education process in mind, just because there's always things we didn't keep in mind. So keep training yourself, continue your education, but really be prospective and plan the types of analyses. What is this? Is it hypothesis-driven, data-driven, prediction, causal, and really sort of staying in the lane of what you're trying to do. Publication, again, publish everything, treat everything along the whole path as if you're going to publish it, so that you can, and so that the barriers are not so high when you do go to that. And your future you even will be happy when you try to augment or build up your own data sets. And also when you're required to share, that will be easier. Share in a fair way so that things are findable, accessible, interoperable, and reusable. And share all, publish all, the raw data, derived data, scripts, containers, software, protocols, etc. I don't know that we have time to get into this, but this is really just the infrastructure of the maintenance of not only data, but the thing we tend, so again, everyone knows how to get a bunch of data, do some analysis, and write their paper. That's fine. The types of things we've been talking about generate provenance and metadata all over the place. So the added layer here, in addition to what we used to do, is get some data, do analysis, compute, do some more analysis, and then write our paper, keep our data locally, write our paper, maybe publish our data publicly. Is all of this generates metadata and provenance information, this IDM representation. So we need to be careful about maintaining our local private metadata. And when you publish, fine, making that not only the data that you choose to make available with your publication, make the metadata that goes with that data in the publication available as well. So the concept here of ReproPonds is just the small local metadata sources versus a Repro Lake, which is the public version of metadata. So that when you can make metadata publicly available, it then becomes much more easy for people to discover and reuse and aggregate at the metadata scale. So again, the tools that we're working on try to make sure that we generate this metadata, that we maintain it locally for you nicely, and then give you a nice pathway to moving that out to the public data sharing sources. And so again, just the importance of local. You may have lots of local private stuff that you haven't made publicly available yet, but there may be lots of publicly available data that is relevant to the question you're asking. Again, this is looking at left hippocampus volume versus age. These blue ones are my local private data, and these red ones come from publicly available ADHD 200 data sets. So again, it's trivial, I say, to add public data sources to what you're doing locally to try to augment and just to advance the ability to look at generalization and look at increasing power and increasing N by accessing public data sources. So the goal is to really make it much more prevalent and easy to make these ReproPubs, these re-executable publications, which include, yes, all of our lovely words that we need, because humans do need to read this and humans do need to understand what we're saying, but we have to have access to the complete data set, to the complete workflow specification. So the data may not have to be publicly available. I know there's private and HIPAA and protections, but someone ought to be authorized to see your data. So whatever the right barriers are for your data, that has to be available to others to access appropriately and associated with the publication. In addition to the workflow specifications, the environment specifications, and the complete results, that when we have all of these bundled together, it makes it so that the complete description is there, and so that anyone can re-execute and therefore more easily generalize or understand the generalization of yours as they add new data or change the approach and things like that. And there's a couple examples that we have that again just make sure that publication that just via supplemental data or references to additional DOI trackable sources for the data, for the workflow, for the execution environment, for the results, and just trying to establish ways of doing that. So I'll stop there. There are so many people that are part of this overall initiative. They're on the slide here, and a number of initiatives that play into this mechanism. And there are links that I see got put in the chat, but are also in these slides as well. And I will make the link to these slides available. And I thank you for listening, both in real time and in the future recordings of this. And I thank the organizers for letting me chat today.
Thank you very much, Professor Kennedy. So I'd like to remind people, if they do have questions, please ask them on the chat. And I guess while people get warmed up, I would like to say thank you again and ask a question of my own, if you don't mind. I really enjoyed this. I think this is like a really, really nice glance on what hopefully the future of publications are going to be, as you said in the beginning, not only for neuroimaging, but for all fields in science. But then it brings me the question, I think we know, or I mean, from what you're saying, from what you're showing, it seems the technological stack is there to have a much better publication system, but we still have the human factor to take into account, right?
Yep.
How are you seeing that people are accepting? Because there is a lot to learn, right? Like we spoke about version control systems and containers and, environments and so on, right? And the regular biologist is not normally up to date with these things, I would say. Like, how have you been dealing with that?
So the joke answer is we need to wait for the old people to retire. Because in generalizing, again, this is not the serious answer, but generalizing, our newer, younger, more Web 2.0 aware folks are much more aware of containers and Git and things like that. But that's not the serious answer. The serious answer was, in part, we need to break this into small things that people can do. And again, if Git for senior biologist X is not going to happen, well, fine. There's other things that we can do. There's tool and making the tools simpler to use. So you can run a tool that wraps the annotation around your standard analysis in ways that are really low barrier. They don't add too much time. They don't add too much conceptual complexity, but yet they give you a version of that data that is sort of has the annotation, has the provenance associated with it. So again, in part, it's making sure the next generation of scientists get this kind of stuff from the get-go, and therefore the barriers are reduced for them because they understand some of these things, and also making the tools some of the tools simple enough that even people who don't had the time to learn too much new stuff can adopt them. So that's kind of the two-headed approach to that, lowering the barriers to using the tools, and also trying to train the next generation of data analysts, so that even if you are a senior person who doesn't necessarily get this, you have your postdoc or your RA can be taught these things and will be happy to learn these things and adopt them. Again, little bits at a time. This is not all going to happen. Don't take the whole. That's why again we got daunted by showing all the things you can do. Pick any one of them, do it, and pick the easiest ones first. And then you'll get some comfort, and then the next harder one.
Okay, great. Sounds like a good approach. I'm happy because I mean, like as is often the case, right? Like normally, like the human factor is what makes or breaks things.
And we used to use the analogy of infecting people. Infection is bad. So nowadays, so now we're trying to inoculate labs. So again, it's one thing for an individual investigator to do this. It's another thing if your lab has the culture of doing this, or if you can get a group of people to go through the same transitional pain that you are. So again, in our approach is it's hard to get one lab to do these, but if we can get a center, an imaging center, to install the certain infrastructure and to just acquire the data off their scanner, with this stuff just there for you. So again, it's different people have different skills and have different tolerances for these, and just trying to make sure that we get to the people who have the tolerance, for some of these, and can make it easier then for the downstream folks to take advantage of it without even knowing it.
Cool. Great. Okay. So there is a question from the audience, from Miguel. So the question is, did you consider creating a score that gets published about how reproducible a paper is? by, for example, checking different items that need to be part of the paper?
Yes, to some extent. We have a publication recently. I'll have to think for a moment and I'll send the link to it. I don't know how much I can co-process here, but essentially taking the autism literature, taking a couple of years of autism publications, we went back through 50 of the imaging publications and tried to assess, okay, if you wanted to find the data, does this paper tell you where the data is? If you wanted to do the analysis, does this paper tell you, what the tools was? So the good news is almost all software, almost all analysis paper says what tool they use. Great. Very often, they rarely tell you what version they use, and they almost never tell you what operating system the analysis was running. If they've generated some results, they make the results available. Mostly no, but occasionally yes. And I don't know, are the statistics well described in terms of what software is used, what covariates were used, and how those were applied. So we did sort of come up with these sort of 4 factors: the data, the software, the environment, and the statistics about how sort of re-executable they are. And I will try to find that and share that with you. The first author is Hodge, and it was F1000 research earlier this year, I believe.
Sounds good. It's interesting because we have something similar in hardware. There is an open O-meter that measures like how open a certain hardware is in those terms. But yeah, it's interesting to see. Is the paper that you mentioned called An Assessment of Autism Neuroimaging Literature for the Prospects of Reproducibility? Okay, we have the link here, and we can share on the chat for people. So if people have questions, they can ask them now. Also, I have a question about, actually, you mentioned that FreeSurfer and Ant have 2 very different results, right? On that front, and I'm guessing they're both open source tools for modeling regions of the brain or for measuring volumes in the brain.
Yes.
And so my question is, do you think it would make sense for the community to come together and say, okay, let's get together and define like not just one of them, but let's maybe get together and decide that we're gonna invest all of our resources in one tool and basically make that de facto like golden standards, like a little bit like an hourglass model, right? I mean, does that make sense at all?
It makes sense, and I exactly understand why one would ask that question. The counter question is there are sort of fundamental differences in the modeling and the assumptions and they're both big enough, historical, well-established tools that I don't think just the practicality of anointing a winner or not is 1, impractical, and 2, may or may not be right. And the question of and brain anatomy is challenging enough that we don't even really know what the gold standard is. So we don't know who is right or not. So we can get a bunch of people together and we will not solve in a 1 week conference a right or wrong. These are different approaches. And so where I think we get better traction is compute power is relatively cheap in the grand scheme of things, and I don't think we need to anoint a winner. We need to anoint use all tools, and look at consistency. There's a specific overlap. Staple is the term that's coming to mind. I can't remember what it stands for, but really doing doing all the approaches and seeing where and when it matters and exposing that, as opposed to cutting off one or another of the lines of analysis when we don't really have a gold standard ground truth.
Okay, thank you. I think that's a quite interesting view on it. Yeah, I hadn't considered that, so thank you.
And while I point out the structural examples because of FreeSurfer and Ant, and there's other tools as well, this is also happens all the time on the functional MRI side of things. There's at least 3 big programs, FSL, AFNI, and SPM. And again, there's just subtle differences. There isn't a right or wrong. There's all sorts of different cultural things that, again, I don't think it would be hard to pick one tool. And it's very hard to pick who's right or wrong, but doing them and looking at consistency, yeah, I think is the same answer there. So that feels like the more practical, pragmatic way to approach this.
Great. Sounds good. So I don't see any more questions from the audience, not on YouTube as well. And I guess if you don't have any further comments, I guess this brings us to the end of our session today. And from us in Open Neuroscience, I would like to thank you very much for taking the time to present this to us. This is super interesting. And thanks for the audience who joined us today. And for those of you who are watching on YouTube as well. So thank you very much, everybody. And with this, I would like to close our session. Thank you.
Thanks, and everyone should just pick at it a little bit at a time. We have office hours. We're happy to help. If we can convince you want to try to do something, we can also be there to help you try to do it. So feel free to contact us, and good luck, making steps in this area.
Great, thanks.